Hello students, in today's module we'll be learning about photosynthetic pigments. Photosynthesis occurs in a variety of bacteria and in unicellular eukaryotes as in blue-green algae as well as in plants. Although the process in these organisms differs in detail, the underlying mechanisms are similar. And much of our understanding of photosynthesis in vascular plants is derived from studies of simpler organisms. The overall equation for photosynthesis in vascular plants describes an oxidation-reduction reaction in which water donates electrons as hydrogen for the reduction of carbon dioxide to carbohydrate. Photosynthesis in plants encompasses two processes: the light-dependent reactions or light reactions, which occur only when plants are illuminated, and the carbon assimilation reactions or carbon fixation reactions. In photosynthetic eukaryotic cells, both the light-dependent and the carbon assimilation reactions take place in the chloroplasts. These are membrane-bounded intracellular organelles that are variable in shape and generally a few micrometers in diameter. Like mitochondria, they are surrounded by two membranes, an outer membrane that is permeable to small molecules and ions, and an inner membrane that encloses the internal compartment. This compartment contains many flattened, membrane-surrounded vesicles or sacs, the thylakoids, usually arranged in stacks called grana. Embedded in the thylakoid membranes, commonly called lamella, are the photosynthetic pigments and the enzyme complexes that carry out the light reactions and ATP synthesis. The stroma, which is the aqueous phase enclosed by the inner membrane, contains most of the enzymes required for the carbon assimilation reactions. Now let's look at the photosynthetic pigments and light absorption. Photosynthetic organisms contain light-absorbing molecules called pigments. These photosynthetic pigments absorb only specific wavelengths of visible light while reflecting others. The set of wavelengths absorbed by a pigment is its absorption spectrum. Different photosynthetic organisms have a variety of different pigments, so they can absorb energy from a wide range of wavelengths. Visible light is electromagnetic radiation of wavelengths 400 to 700 nanometer, a small part of the electromagnetic spectrum, ranging from violet to red. The energy of a single photon, which is a quantum of light, is greater at the violet end of the spectrum than at the red end. When a photon is absorbed, an electron in the absorbing molecule, we call it chromophore, is lifted to a higher energy level. This is an all or nothing event. To be absorbed, the photon must contain a quantity of energy that exactly matches the energy of the electronic transition. A molecule that has absorbed a photon is in an excited state, which is generally unstable. An electron lifted into a high energy orbital usually returns rapidly to its normal lower energy orbital. The excited molecule decays to the stable ground state, giving up the absorbed quantum as light or heat or using it to do chemical work. In photosynthesis, there is a direct transfer of excitation energy from an excited molecule to a neighboring molecule. Just as a photon is a quantum of light energy, so the exciton is a quantum of energy passed from an excited molecule to another molecule in a process called exciton transfer. Let's look at the absorption of visible light by photopigments. Plants are green because their pigments absorb light from the red and blue regions of the spectrum, leaving primarily green light to be reflected or transmitted. Let us observe and compare the absorption spectra of the pigments with the spectrum of sunlight reaching the Earth's surface. The combination of chlorophylls, that is A and B, and accessory pigments enables plants to harvest most of the energy available in sunlight. The relative amounts of chlorophylls and accessory pigments are characteristic of a particular plant species. Variation in the proportions of these pigments is responsible for the range of colors of photosynthetic organisms, from the deep blue-green of spruce needles to the greener green of maple leaves to the red, brown or purple color of some species of multicellular algae and the leaves of some foliage plants favored by our gardeners. There are three major types of photosynthetic pigments, namely chlorophyll, carotenoids, and phycobilins. Chlorophyll pigments. The most important light-absorbing pigments in the thylakoid membranes in chloroplasts are the chlorophylls, green pigments with polycyclic planar structures resembling the protoporphyrin of hemoglobin, except that magnesium, not ferrous, occupies a central position. The four inward-oriented nitrogen atoms of chlorophyll are coordinated with the magnesium ion. All chlorophylls have a long phytol side chain, esterified to a carboxyl group, substituent in ring 4. And chlorophylls also have a fifth five-membered ring not present in heme. The heterocyclic five-ring system that surrounds the magnesium ion has an extended Pauline structure with alternating single and double bonds. Such Paulines characteristically show strong absorption in the visible region of the spectrum. The chlorophylls have unusually high molar extinction coefficients. Therefore, they are particularly well suited for absorbing visible light during photosynthesis. Chlorophyll A is the most abundant pigment in plants. Chlorophyll A absorbs light with wavelengths of 430 nanometer, which is the blue range, and 662 nanometer in red. It reflects green light strongly, so it appears green to us. It contains a hydrophobic, fat-soluble phytol chain that allow it to be embedded in a lipid membrane. Chlorophyll B has a structure similar to that of chlorophyll A. It absorbs light of 453 nanometer and 642 nanometer maximally. It is not as abundant as chlorophyll A and probably evolved later, but it helps increase the range of light a plant can use for energy. Chloroplasts always contain both chlorophyll A and chlorophyll B. Since their absorption spectra are sufficiently different, they complement each other's range of light absorption in the visible region. Most plant contains about twice as much as chlorophyll A as chlorophyll B. The pigments in algae and photosynthetic bacteria include chlorophylls that differ only slightly from the plant pigments. Chlorophyll is always associated with specific binding proteins, forming light harvesting complexes, we call them LHCs, in which chlorophyll molecules are fixed in relation to each other, to other protein complexes, and to the membrane. The detailed structure of one light harvesting complex is known from X-ray crystallography. In light harvesting complex LHC2, the functional unit is an LHC trimer with 36 chlorophyll and 6 lutein molecules. Shown here is a monomer viewed in the plane of the membrane with its 3 transmembrane alpha helical segments, 7 chlorophyll A molecules, 5 chlorophyll B molecules, and 2 molecules of the accessory pigment lutein, which form an internal cross brace. Now let's look at carotenoids. In addition to chlorophylls, thylakoid membranes contain secondary light-absorbing pigments or accessory pigments called carotenoids. Carotenoids may be yellow, red, or purple. The most important are beta-carotene, which is a red-orange isoprenoid, and the yellow carotenoid lutein. The carotenoid pigments absorb light at wavelengths not absorbed by the chlorophylls and thus are supplementary light receptors. There are two types of carotenoids, carotenes and xanthophylls. Carotene is in orange color while xanthophylls are yellow in color. These accessory pigments protect the chlorophyll against photooxidation and then transfer light energy to chlorophylls. It also helps in preventing photodynamic damage. Also, carotenoids are able to express themselves in mature leaves even if chlorophyll begins to disintegrate. Now let's study the phycobilins. These photosynthetic pigments are open-chain tetrapyrols which lack magnesium ion and the phytol tail. Green algae and red algae possess phycobilins such as phycoerythrobilin and phycocyanobilin respectively as their light-harvesting pigments. Phycobilins are covalently linked to specific binding proteins, forming phycobiliproteins, which associate to form highly ordered complexes called phycobilisomes that constitute the primary light-harvesting structures in the microorganisms like green algae and red algae. So let's look at a phycobilisome. In these highly structured assemblies found in cyanobacteria and red algae, phycobilin pigments bound to specific proteins form complexes called phycoerythrin, PE, phycocyanin, PC, and allophycocyanin, AP. The energy of photons absorbed by PE or PC is conveyed through AP, which is a phycocyanobillin binding protein, to chlorophyll A of the reaction center by exciton transfer. Phycobiliproteins then pass the light energy to chlorophylls for photosynthesis. The phycobilins are especially efficient at absorbing red, orange, yellow, and green light wavelengths that are not well absorbed by chlorophyll A. They can be found in different combinations attached to phycobiliproteins. Experimental determination of the effectiveness of light of different colors in promoting photosynthesis yields an action spectrum, often useful in identifying the pigment primarily responsible for a biological effect of light. By capturing light in a region of the spectrum not used by other organisms, a photosynthetic organism can claim a unique ecological niche. For example, the phycobilins in red algae and cyanobacteria absorb light in the range 520 to 630 nanometer. So, this allows them to occupy niches where light of lower or higher wavelengths has been filtered out by the pigments of other organisms living in the water above them or by the water itself. Now, let's study the role of photosynthetic pigments in photosynthesis. The light-absorbing pigments of thylakoid or bacterial membranes are arranged in functional arrays called photosystems. In spinach chloroplasts, for example, each photosystem contains about 200 chlorophyll and 50 carotenoid molecules. All the pigment molecules in a photosystem can absorb photons, but only a few chlorophyll molecules associated with the photochemical reaction center are specialized to transduce light into chemical energy. The other pigment molecules in a photosystem are called light harvesting or antenna molecules. They absorb light energy and transmit it rapidly and efficiently to the reaction center. The chlorophyll molecules in the light harvesting complexes have light absorption properties that are slightly different from those of free chlorophyll. When isolated, chlorophyll molecules in vitro are excited by light. The absorbed energy is quickly released as fluorescence and heat. But when chlorophyll in intact leaves is excited by visible light, very little fluorescence is observed. Instead, the excited antenna chlorophyll transfers energy directly to a neighboring chlorophyll molecule, which becomes excited as the first molecule returns to its ground state. The transfer of energy, which we call exciton transfer, extends to a third, a fourth, or subsequent neighbor until one of a special pair of chlorophyll A molecules at the photochemical reaction center is excited. In this excited chlorophyll molecule, an electron is promoted to a higher energy orbital. This electron then passes to a nearby electron acceptor that is part of the electron transfer chain, leaving the reaction center chlorophyll with a missing electron. We call it an electron hole, denoted by a plus sign. The electron acceptor requires a negative charge in this transaction. The electron lost by the reaction center chlorophyll is replaced by an electron from a neighboring electron donor molecule, which thereby becomes positively charged. In this way, excitation by light causes electric charge separation and initiates an oxidation-reduction chain. So that's all we'll be learning in this module. The subsequent reactions involved in photosynthesis will be discussed in a separate module. So to conclude, in photosynthesis, light energy is captured by photosynthetic pigments. These photosynthetic light-absorbing pigments are found in bacteria, blue-green algae, and photosynthetic plants. There are three major types of photosynthetic pigments, namely chlorophylls, carotenoids, and phycobilins. These photosynthetic pigments absorb only specific wavelengths of visible light while reflecting others. The presence of a combination of chlorophylls, A and B, and accessory pigments enables plants to harvest most of the energy available in sunlight. So the relative amounts of chlorophylls and accessory pigments are characteristic of a particular plant species. Variation in the proportions of these pigments is responsible for the range of colors of photosynthetic organisms. And in the light reactions of plants, absorption of a photon excites chlorophyll molecules and other accessory pigments, which funnel the energy into reaction centers in the thylakoid membranes. Thank you.